We're going to look at what you need to know about DNA for the higher biology course. We're going to start off by looking at where DNA is found in the different types of cells that we're going to study. We're going to start off by looking at bacterial cells. Bacteria are all around us, but they're too small to see. In this petri dish here, we have bacteria growing, but if we look at the arrow, it's pointing at one of these single dots. That's actually a colony of bacteria. There's possibly millions of bacteria in that colony, and they've all grown up from a single bacterial cell. So you can see many of them, but you can't see single bacteria. You would need a microscope to view a single bacteria. Here's some examples of different types of bacteria we can get. And if we look at the size here, I've drawn to scale a 1 micrometer. Okay, so you would get 1,000 micrometers in a millimeter, so it gives you an idea how small the bacteria are. Approximately about 1,000 would stretch across in 1 millimeter. On the top left there, we've got Staphylococcus aureus, and those are bacteria that you can find on your skin. They're quite commonly found on skin. They can sort of be harmless on your skin, but if they get into an infection, if they get into a cut, they can cause an infection. And if they go onto food, they can cause food poisoning. They give off toxins if they grow up on food. Down in the bottom left there, we've got group A Streptococcus. This group of bacteria can cause many different types of infections, from minor illness to very serious deadly diseases. Some examples of diseases they cause are scarlet fever, strep throat, and cellulitis, which is a skin infection. On the top right there, we've got Vibrio cholerae. They have a flagellum, that's the sort of tail that you see that allows them to move around. Some strains can cause cholera if you drink contaminated water or eat contaminated food. And some of the symptoms of cholera can be vomiting, diarrhea, and dehydration. And on the bottom right there, we've got Bacillus anthracis. This causes anthrax, which is a common disease of livestock, and it can occasionally affect humans. So that's some examples of bacterial cells. If we zoom in now and look at the Vibrio cholerae bacteria, and we'll look at the parts of this bacterial cell. So bacteria are classed as prokaryotes. You can see that at the top of the screen there, prokaryotes. This means that they evolved before they had nucleuses, so before the nucleus. And so you don't see any organelles in a bacterial cell. We don't have a nucleus, we don't have mitochondria, we don't have chloroplasts. So everything's sort of loose here in the middle of the cell. So the DNA is found in a circular chromosome. And there's also some DNA found in plasmids. You looked at plasmids in National 5 when you did genetic engineering. So that's the places you would find DNA in a bacterial cell. DNA, as we'll discover, is involved in protein synthesis, in the making of proteins. And we'll find that ribosomes are really important in that. So we've also got ribosomes here in the bacterial cell. Other things we've got is the cell wall around the bacteria. Okay, and this is not made of the same cellulose that you get in the plant cell wall. So it's different structure and composition, but you do get a cell wall in the bacteria. We don't need to learn what it's made of. Okay, these are the types of cells we'd find in humans, and they're also eukaryotic cells. This means they actually have a nucleus, okay? Slightly more advanced design of cell, a bit more organization. Can you think of what other types of organisms would be called eukaryotes? What other ones have we to study at higher biology? Okay. Did you think of plant cells? And the other one is fungi, fungal cells. are eukaryotes. And the fungus that you probably come across studying at higher biology is yeast. That's the one we'll most commonly come across. Okay, so let's look at the organisation here in this cell. So a bit more organisation. We've got mitochondria here. Okay, these are organelles in the cell, mitochondria. What process, is a well-known process in biology, but what process takes place in mitochondria? Okay, did you think of respiration? Hope you did, good. Okay, what is the purpose of respiration for a cell? Okay, did you think of to make energy for the cell? And when it's made that energy, do you know what form the energy is stored in? How is the energy stored to be passed on to other parts of the cell and be used? Okay, did you think of ATP? Yeah, so that's how energy is stored. Right, so mitochondria have got all of the enzymes, or a lot of the enzymes to do with respiration are in the mitochondria. Some of them are also in the cytoplasm. But basically, if you have all the enzymes in one place, that helps the organisation and helps the processes happen. Okay, so here's another organelle, the nucleus. And it has the chromosomes in it. Okay, so the chromosomes contain the DNA. Okay, DNA, which has all the information for the cell, the genetic information for the cell. And the DNA in a eukaryote is on linear chromosomes. Okay, can you see here they're like little lines. They come in matching pairs of chromosomes, but they're in lines. And the DNA is wound tightly around histone proteins. Okay, so it's wound up, and it can be unravelled again when we're copying it. But that I've just really drawn it like that, but it would probably normally be sort of clumped up and wound up. So that's your DNA in the nucleus. If you notice, we've got a double membrane around the nucleus. Okay, we also had a double membrane on the mitochondria, but double membrane around the nucleus, and it has gaps in it, pores in it. If we can think of the messenger RNA can come out these pores and go to the ribosomes. So in this cell, we've got little ribosomes drawn as dots. Okay. You can also get ribosomes on this stuff here, which is called endoplasmic reticulum. Don't need to know about that really for higher. It'll come up in advanced higher biology if you do that. But you'll quite often see these things drawn on cells. And we've also got another thing here called Golgi apparatus. Okay, here. So that's your endoplasmic reticulum and Golgi apparatus. Don't worry about those at the moment. I've just drawn them there to make the cell look more complete. The ones you really need to remember: cell membrane, nucleus. Know that the chromosomes are linear chromosomes. We've got mitochondria and we've got ribosomes. Now, another interesting thing is we also have a small amount of DNA somewhere else in the cell. Do you know where that would be in a human? So we've got linear chromosomes in the nucleus, but where else might you get a little bit of DNA, or you do get a little bit of DNA? Did you think of the mitochondria? Okay, the mitochondria has a tiny amount of DNA. It's probably got the information for enzymes involved in respiration on it. But interestingly, this DNA is actually inherited from the mother's line. So it's your mother's mother's mother's. You've inherited that down your family tree from your mother's mother's mother onwards. And so it can actually give us some interesting family tree information. You can go back and find your ancestry on that line if you were to study that mitochondrial DNA. Because it's been inherited on the egg cell. Okay, that's an interesting bit. So we have linear chromosomes with DNA, and we have mitochondria with DNA. Plant cells also have DNA where Okay, did you think of chloroplasts? Okay, they have DNA in the chloroplasts. And fungal cells, how do they have their DNA organized? Okay, right, so in yeast, it could also have plasmids. Okay, so that's another thing to remember. Okay, so that's everything I think about where DNA is found in the cells that we need to know for higher biology. If we zoom in on the nucleus of a human cell, we'll see that there are chromosomes, and the chromosomes come in pairs. So the DNA is found on the chromosomes, and along the chromosomes we have the genes. Okay, so the level of organization is DNA, genes, chromosomes. When people were studying the chromosomes, they actually added dyes to them, and we can see that there's different banding there of where the dye attached to the chromosomes, and that was to do with how many A and T or G and C bases there was on particular sections of the chromosomes where the dyes added. So it helped people doing the early research on chromosomes to match up and notice that they came in pairs. Okay, and I suppose it's a bit of an idea that there's different genes along the chromosomes there. Okay, so the DNA has been tightly wound around proteins in order to pack it into these chromosomes. Can you remember what name I said the proteins were called that the DNA gets tightly wound around? Okay, the proteins are called histones. You need to know that word for higher biology. The DNA winds around histones proteins when it goes into the chromosomes. Okay, so humans have got 23 pairs of chromosomes. In this particular cell, it's got, bottom right there, you can see there's an X and an X chromosome. That's what you would get in a human female cell. Okay, if it was a male cell, you would have an X and a Y, and the Y would be a smaller chromosome. They're not quite an exact match at all, whereas the two X chromosomes here match up. But all the others, the 1 to 22, they come as exact sort of matching pairs. Okay, so that's the sort of normal human cell. You can get some genetic disorders where you might have different numbers of pairs or of chromosomes and things. The whole set of the chromosomes is called a karyotype. You don't need to know that word either, but that's the name for that, the whole set of chromosomes in a human cell. So we're going to move on and just have a look at a bit more detail about what is the structure of a DNA molecule. So DNA is made up of nucleotides. Here is one nucleotide. And what we see here is we have in the middle of it the blue deoxyribose sugar. Okay, so a sugar is a molecule. It's a carbohydrate. It's got carbon, hydrogen, and oxygen making it up. We don't need to know the details of the molecule, so it's just represented here. It actually has an oxygen at the top there, though. That would explain why there's not a number allocated to that, whereas the other corners of it are all carbons, and we have a numbering system for them. So this right hand one is carbon 1, then bottom right carbon 2, carbon 3, important one to remember, carbon 4, and then carbon 5 has a phosphate attached to it. Okay, it's got a phosphate group attached to it. And carbon 1 has a base attached to it. Okay, and carbon 3 is just right on the deoxyribose sugar. Okay, so if we remember that the bases are adenine, thymine, guanine, and cytosine. The A goes with T, remember this from National 5 as well. A goes with T, G always goes with C. One way to remember this is at Glasgow Central. I like that way of remembering it. Okay, so at Glasgow Central. We're also going to find out we have the 5' and the 3' end, and then the 3', 5' end. They're going in opposite directions. We're going to find out a bit more about that in the next slides. Just bring that up. So let's zoom in and look at, okay, so here is the DNA molecule visualized in detail. So again, here we have the top left there, we've got the nucleotide and it's got the base G on it. It's got the deoxyribose sugar and the phosphate group at the top, you know, at the top there. Okay, that's the phosphate and the deoxyribose sugar. So what we have with the blue background here, we've got the sugar phosphate backbone we talked about, right? So the sugar phosphate backbone of that strand, and there's the same going down the opposite side, and we've got the bases between the two backbones. So we've got the two backbones of the DNA and the bases in between. Okay, so again, the numbering system, the top left there, the phosphate group is actually on the 5' carbon, the 5 carbon of the sugar. So that's why it's called the 5' end of the DNA, that. So it's got the phosphate group on the 5' end, and if we look at the bottom left, that is the carbon on that deoxyribose sugar that's finishing off that chain. So it's on the bottom left, it's the 3'. Okay, and what we find is when new nucleotides get added on, they always get added on to the 3' end. So it's really important to remember that. The 3' end is straight onto the deoxyribose sugar. And the other strand runs in the opposite direction. It's called the antiparallel strand. So in that one, the phosphate's on the bottom right there, on the 5' end of the phosphate's bottom right, and the top is the 3' end. Okay, you can see the bases matching up here, G and C, A and T. And do you know what type of bonding goes between the bases? Have you done that yet? Okay, did you think of hydrogen bonding? It's actually hydrogen bonding. And if you've done chemistry at all, you'll know that, maybe know that it's quite a weak type of bond. Okay, so it holds the two sides of the DNA together, but they can also be easily separated. The two strands can be easily separated. And what we find is that that's needed when we want to copy DNA. So you can copy it into two strands and then make a copy of each side. We end up with two DNAs. That happens in DNA replication. But at the moment, we're just looking at the structure here. So down the middle, between the bases, we've got hydrogen bonding, that's weak bonding. Down the backbone, it's a stronger type of bonding. If you were to go to chemistry, that's actually called covalent bonding, much stronger. So the backbone's more likely to stay intact. Right, what next? Let's just see what other diagrams I had here. This was zooming in to look at a bit more of the sort of chemistry of it, just to look at. So again, top left, don't worry about the chemistry here, it's just to show you what it would look like if you happen to be doing chemistry. It'd make more sense maybe. Top left there, we've got the phosphate group. If you were to look up, I think I've drawn an extra minus on there actually, but it should be just 1 minus on the, so it's PO4 minus. If you look in the data booklet for a phosphate group, it's straight onto the deoxyribose sugar, then another phosphate, deoxyribose sugar, phosphate, deoxyribose sugar going down that backbone. The backbone of DNA, or DNA actually, is a negatively charged molecule because of all those phosphate groups, and that comes in when we start to look at sequencing DNA or pulling it through a gel. You've got the positive and negative electrodes, and the DNA will always go towards a positive electrode. You can see the hydrogen bonding on there between the different groups. So you can see that the A and T, I think, has the 2 hydrogen bonds, and the G and C has got the 3. Okay, I hope I've got that the right way around. But anyway, you can see that they match up, don't they? They've got to match up with their hydrogen bonding. I may have that the wrong way around, but you don't need to know that. That's just looking at the detail of the chemistry there. Okay, so I hope that's helped you understand a bit more about the structure of DNA. So that's us reached the end of looking at key area 1, which is the structure of DNA. One final thing that we need to remember is, so looking back at this diagram of the molecule, is actually that it forms a double helix. So you see this, this sort of, it looks like a spiral staircase sort of structure. Okay, so that is called the double helix, and here I'm just using the colours to represent the A and T and the G and C. This is okay. So what happens is we have a code. So the basis, the code, the sequence of bases is called the genetic code on the DNA. And in the next 2 sections of the course, what you're going to find out about is how we convert that code, the sequence of bases on the code, into the sequence of amino acids in a protein. Okay, and so the code in the DNA is really important to how the cell works and what proteins are in the cell. So the base sequence of DNA forms the genetic code. Okay, the DNA was made of nucleotides, which were, can you remember what was in a nucleotide? Okay, so the nucleotide's made up of your phosphate group, your deoxyribose sugar, and your bases. Okay, so you've got your nucleotides, and there's the 4 types of bases in the DNA. A goes with, which one did it go with? A goes with T, G goes with C. Okay, and we're going to find out that it's every 3 bases that is the code. The code is, it sort of goes in 3s. So that's an interesting thing you'll find out in the next bit. And the structure is the double helix. That was discovered by, well, there's 4 scientists that are particularly remembered for discovering the structure of the fact it was the 2 strands. So Watson and Crick and Wilkins and, oh, sugar, what's her name again? Forgotten her name. Rosalind Franklin, famous women scientist that I forgot the name of temporarily there, but she had done the X-ray image that actually revealed the sort of information for the structure of DNA. But unfortunately, she was a famous protein chemist, but perhaps with the overexposure to X-rays in her career, she actually died fairly young of cancer. And actually the Nobel Prize was then awarded to Wilkins, Watson and Crick, so she missed out on being awarded the Nobel Prize. And also they don't award Nobel Prizes posthumously when somebody's died. So the DNA, that was the structure of DNA in the double helix. Some other early research was done by Shargarth, where he matched up and discovered that the bases went together. So he noticed there was a particular ratio between the A and T and the G and C, so that was quite interesting. But so we've learned lots about the structure of DNA, okay, and you're going to go on and look at how that then becomes, so the genetic code in the DNA, how does that make the proteins in the cell? Okay, so good luck with learning that. So I think that's 20 minutes we've done on basic structure of DNA, covered everything that's needed, I think.